Okay, so we've looked at DNA, its structure, and the structures that it's involved in, specifically chromosomes. But now we need to move on to what DNA actually does. And DNA does two things. Its ultimate goal is to help instruct the cell to do all its important processes. But before it can do that, or before we can consider that, we need to talk about how DNA replicates. So let's cast our minds back to what genetics is all about. It's about inheritance. It's about parents passing on their information to their offspring. And to do that, a couple of things have to happen. First of all, DNA has to be copied. Well, if we have a recipe book, like I've been talking about before, and I pass that on to my son, I don't lose all that much. I can't make chocolate chip cookies the same way, but I'm not going to die. But in the case of a cell, if any cell passes on its DNA, it cannot do the jobs of life. It will die. So DNA has to be copied, and the parent has to keep one copy for themselves and pass the other copy on to their offspring. Now remember, we're talking here about the level of cell reproduction. Don't worry about whole human reproduction yet. We're going to talk about how multicellular organisms' sexual reproduction happens later on. We're talking about cells, and the inheritance is between one cell and another cell. So first of all, the DNA has to be copied, and that copying process is known as replication. And the ability to replicate is the thing that gives DNA its remarkable ability to be the material of inheritance. Okay, well, once the DNA is replicated, the copies have to be put into separate cells. Now, we're only going to cover the process of replication in this lesson. The process of cell division is going to come in a later lesson. So don't worry about the next step. Just think here about how does DNA go from being one molecule to being two identical photocopied molecules. And by photocopying, again, I'm using that book analogy that we've been using in the last couple of lessons. Now, here is a short video which summarizes pretty much everything that I am going to say, and I recommend that you actually watch it before listening to the rest of this lesson. You can find the link and you can follow the link from the PowerPoint version. Now, you don't have to, but I think that if you see the video first, then what I'm going to say is going to make more sense. All right. Well, we have a strand of DNA, and the first thing that has to happen is that the two strands, the two sides, have to separate. And that separation is caused by an enzyme called helicase, which breaks those hydrogen bonds between the bases on the neighboring strands of DNA. So here we have one strand, so the template strand and the complementary strand. Here are the nucleotide bases matched up with the base pairing rule and held together by hydrogen bonds in between them. So a helicase is going to come in and is going to break those weak hydrogen bonds and enable the two strands to separate. The region where this split is happening is called the replication fork. And here is another diagram of DNA. And you can see there is the replication fork, the place where the double strand is split into two separate strands. And here is where helicase will fit, this enzyme which is causing those two strands to separate, to unravel from each other. Well, after separation, the nucleotide bases have a free end. They're not bonded to their opposite partner anymore. They are free. And so they can now bond with nucleotide bases which are not in a strand, just floating around in the cell, but to make that happen, to make that bonding process happen, requires some complicated steps and a bunch of other molecules, including a bunch of other enzymes. So we're now going to talk about how those free nucleotides floating around in solution in the cell get bonded together into a new strand. Well, we've had strand separation, and the next thing that's going to happen is that a short section of RNA, remember that's another molecule very similar to DNA, and again we'll talk about its structure in another lesson. But a short section of RNA called a primer is constructed by an enzyme primase. So primase is an enzyme. Remember, enzymes speed up reactions but are not used up by the reaction. And primase puts together pieces of RNA to create this thing called a primer. And the primer then attaches or gets attached to special sections of the DNA that are ready to receive the primer. Well, the primer doesn't do anything itself, but it allows another enzyme called DNA polymerase to attach to the DNA strand. You can think of each strand as being a railway track and the DNA polymerase as being a train on the track. And the primer is something that allows the train to find the track and fit onto it. Now, notice this word DNA polymerase. That's one of those classic big biology, fancy technical words, but we can break it down and make it much more memorable. Well, it's DNA polymerase because it's got something to do with making new DNA. It's got ASE at the end, and we know that every time you see ASE, it means it's an enzyme, and it's something to do with a polymer. Well, remember, a polymer is a long chain of smaller units joined together. So this enzyme is one that makes the polymer of DNA. That's all the word means. And I think that helps us remember it. Well, DNA polymerase is now attached to the original strand, and it helps bind those nucleotides that are just floating freely in the cytoplasm to bond to their complementary bases. I'm going to show you some more images in a minute that I think will help you understand or follow this idea of DNA polymerase bonding these free nucleotide bases into a new strand. But before that, I need to make a note about direction. Nucleotides, these free floating nucleotides that are now going to get attached and made to create a new DNA strand, are always added in a particular direction. And if you remember back to videos ago, you'll remember that we named the numbers of carbon atoms in the deoxyribose sugar ring 1, 2, 3, 4, 5, or actually 1 prime, 2 prime, and so forth. And that means that each backbone of DNA has a direction. It's not symmetrical with, it doesn't go, if you look at it from one way, it's actually slightly different than if you look at it the other way. And so nucleotides are always added in the direction that goes from the 5 prime carbon atom toward the 3 prime carbon atom, or in the 5 to 3 direction. Another way to say the same thing is at the front of a developing new strand of DNA is always the 3 prime end. Now that's going to sound really weird. Before I give you some illustrations of how that works, here's a little memory device that works for me. Students look forward to 5 to 3. Well, 5 to 3 is just before the bell goes for home time, right? And so if students look forward to 5 to 3, well, DNA is replicated forwards from the 5 prime end towards the 3 prime end. And I think that that's a useful way of memorizing which direction DNA synthesis happens. Here's a visual representation of the same thing. Now, Here is our nucleotide. Remember, a nucleotide is one monomer of the DNA polymer. Here we have our deoxyribose sugar. Here is our phosphate group. Here is our nucleotide base. This one isn't an adenine, but it could be a thymine, a cytosine, or a guanine. It doesn't matter. And if you remember back to the lesson about the directionality of DNA, this is the 5' end because there's your 5' carbon, and this is the 3' end because there's your 3' carbon. All right, let's simplify that. And here we have the whole nucleotide, one single nucleotide simplified. There's your 5' end. And now when they get added from the 5' end toward the 3'. Remember, your 5' carbon atom is here and your 3' carbon atom is there. And the addition is always like that. Okay, that's directionality in DNA synthesis. 5 to 3 direction. All right, because one of the DNA strands has, if you're looking from one end of DNA, and you'll see that one of the strands from where you're standing is going from 3' to 5', and the other strand, the opposite strand, the complementary strand, is going 5' to 3'. And because of that, slightly different processes are needed for replication. Now, for this course, you don't need to know all the details about the different processes by which those two strands get replicated. The reason I'm telling you is that as soon as you go away from this PowerPoint and you start reading any textbook or looking at any information on the internet, you're going to find information about leading strands and lagging strands, about Okazaki fragments, and a whole bunch of other molecules and enzymes, which are not required for this course. So I've left them out, and I'm only trying to keep the stuff that is essential for you guys to know. But you will find more information, and it's helpful for you to be aware of it, but it's not essential for you to remember. All right, I've only included one Pearson question because at this point, there is only one question that you guys would be able to answer. We need to get some more information behind us before we can answer more questions in Pearson. There is your textbook chapter and the pages that have discussed what we have talked about here, the replication of DNA. And here are two videos you might want to look at. If you recall from a previous lesson, I said that this is that if you wanted to watch the crash course lesson on DNA, that you should just watch it up until about 8 minutes 53, because beyond that, the information is to do with replication. So you can now finish that video. And you also might look at Amoeba Sisters because they also do a good job. In both of these cases, you will find more information than we have covered. Remember, what I have covered is necessary for you to remember, but you will probably find it beneficial to listen to the other stuff because it fills in some of the gaps that I've left. All right, folks, that ends replication for today.